Hello and welcome to Chapter 19, Introduction to Molecular Genetics. So we're going to be talking about DNA and all of our genetics in this chapter. So we're going to describe the makeup of nucleosides, nucleotides, oligonucleotides, and polynucleotides. We're going to describe the primary structure of DNA and RNA, as well as their secondary and tertiary structures. We're going to explain how replication takes place and describe the roles of our DNA polymerase. We're going to look at transcription and describe the role of RNA polymerase in this process. We're going to name 3 types of RNA, identify their roles in translation now, then explain how E. coli, so it's not just a bad bacteria, control the expression of genes. Given an example of recombinant DNA and how it's used to help humans, and we're going to explain the term mutation in genetic diseases. So let's first look at some mutations here. So if we're going to be looking at our genetic makeup for this chapter, let's start off with looking at what happens if we can change that genetic makeup or create a mutant. This has created a lot of controversy here in the last 15 to 20 years, especially with the company Monsanto, since they are kind of the ones that makes a lot of these herbicides that had a lot of side effects. So they were the first ones to come up with Roundup. So Roundup is a weed killer. But have you ever thought about how Roundup and stuff like it actually only kills weeds and not your grass and not your plants? It only kills weeds? Well, let's take a look. So they were in their factory for this Roundup, and they noticed that in a waste column, so in a kind of a waste area of the plant, they found this strain of bacteria, this Argobacterium. And they found that this bacteria was growing just fine. And so it was insensitive and has this EPSP synthase enzyme that was insensitive to the glyphosate. So it grew just fine. So it had a genetic makeup that could withstand this weed killer. So what they thought up was that they could use this bacteria's DNA. If they could insert it into other plants, then those plants would be all completely insensitive to the Roundup. So it wouldn't be killed by this weed killer. So let's take a look at how they actually did that. So they took this Argobacterium, and it kind of looks like this. And they had to insert its DNA into the plant cell. Remember, bacteria do this. Bacteria can insert their DNA into other cells. So it inserts it into the chromosome. So they're going to leave it in there for about 24 hours. And that medium also that it's in has kind of root growers and helps it grow. So once those roots start growing, start getting shoots of corn, and those shoots of corn then they take and put into another growing medium, and once it's got good enough roots, they can actually plant it. And that new plant that they have now, like we said, now has a genetic makeup that will not be killed by this particular weed killer. So then they can start selling these plants that are resistant to their herbicide, and they can sell the herbicide at the same time. So kind of an interesting model for this company, that they first made the weed killer, and then they made the plant that resisted the weed killer, and then they can sell both of them. So kind of an interesting concept that they came up with. You can understand why that people get pretty irritated with them, since they can sell both of them at the same time. So once these plants started to be produced, they gave them a new term. And this new term was GMO, genetically modified organism. So a lot of companies were saying, oh man, these are now GMOs. They are all bad because they've been modified. Well, just because something's been modified does not mean that it is bad. At this time, there's been no scientific evidence to show that GMO products actually do anything to us. We just still are going to digest the food normally and then pass it through us through our waste. So they're kind of doing a lot of scare tactics for these GMO products and trying to get you to buy non-GMO, but they're not all bad. A lot of times, like we see in the picture, we're using genetically modified organisms to just let us know that we have been able to insert a particular gene into a organism. So particularly like for these rats, a lot of times they'll insert genes they think have an effect for, let's say, diabetes. They want to give these rats diabetes. But how do they know if the gene was actually inserted? We can, see all we want that, or put everything, insert everything, but how do we know that it was inserted? We can't see the DNA ourselves. So what they do is they add on to each of these pieces of DNA they want to insert a green fluorescent protein, so a GFP. And what this green fluorescent protein actually does is it makes the organism fluoresce green. And if it's fluorescing green, they know that whatever gene they inserted was actually inserted into the organism. So in the case of these rats, these are GMO rats. They were able to insert a particular gene in there, and they confirmed it using this green fluorescent protein. So then they can go and use these rats for whatever testing they need to. So if they were going to do diabetes testing on it, well, now they know that these are the correct subjects for them to use. So they can also do this into insects or whatever organism they're looking into. So whatever they want to insert, or if they want to practice inserting a specific type of DNA or to see if it actually does get inserted, they can put it into, this is a fly. Here's another rat. This is a rat with no fur, so the whole thing just fluoresces. Is this not the stuff of nightmares or what? Could you imagine a little rat running around that glows in the dark? It would be a scary Halloween prank. But they could even make it or do it to check out kind of the internal organs of something like this little worm. So they could, because it's glowing, they're actually able to see all of the different organs inside of this worm. So at this point, this is actually pretty primitive. This is kind of pre-CRISPR. So if you've never heard of CRISPR, CRISPR is a way for geneticists to actually much more easily cut and insert kind of whatever DNA they want into our or something's genetics. However, CRISPR doesn't just insert, but it can edit that DNA as well. So it can be very particular. It can go to a very particular spot in a DNA sequence, edit it somehow, either add, subtract, modify what's there, and then leave. Now we saw with the corn a little bit earlier, it was a bacteria that was actually editing the DNA. Well, that's essentially what CRISPR was. CRISPR was adapted from a naturally occurring bacteria that they then took and modified so they could use that same editing system. So kind of some novel ways that they're using CRISPR right now is they're actually trying to edit out kind of the cancer genes from our DNA. The catch is that they can't just like insert CRISPR into our bodies. They aren't just going to like give you a shot of CRISPR. That's kind of risky. So what they're doing is they're actually taking your cells, putting them in a petri dish, applying the CRISPR, editing out the DNA, and then putting it back into patients and seeing what happens. So we kind of live in an interesting time because a lot of these disorders that we haven't been able to cure, like cancer, we may be able to modify our DNA so we are resistant to cancer. There are several organisms like the naked mole rat or elephants and bowhead whales that are resistant to cancer. So they're trying to figure out what DNA they have that makes them resistant to cancer, and then modify ours to be the same. So it'll be very interesting to see what they come up with in the next 10 or 20 years using this CRISPR. But we'll look a little bit more at CRISPR a little bit later. But let's start talking about the actual makeup of our DNA and our nucleic acids. So what are nucleic acids? They are polymers that consist of nucleotide residues. Well, what is a nucleotide? Well, a nucleotide contains 3 parts. The first part is a phosphoric acid. So those phosphor ester bonds that we looked at in chapter 15, they are coming back right now. Then the second part is a monosaccharide. So there's our chapter 16, our sugar residues. And our last one are our organic bases. Now, if you think in biology, if you think of the DNA bases, you think of a base as in like a building block or something that is the lowest aspect of something, basic, right? But that's not actually what they're referring to when they're talking about an organic base. They're legitimately talking about acid or base chemistry. It's an organic base because they have these nitrogens in them. Nitrogens, we know, have a pH that is basic. So these are actually a basic pH that we're talking about. So if you've kind of learned anything about DNA, these bases should be familiar. Our thymine, our guanine, our cytosine, and our adenine. So our A, T, and our C and Gs. So let's delve into each one of these a little bit further. So we said we first had our phosphoric acid in each of these nucleotides. We know that at low pH, we want to add on as many hydrogens as we possibly can. So this is not what it's going to look like because it's got all hydrogens. It's not going to react. However, at physiological pH, so around our pH of 7, we lose 1 or 2 of those hydrogens. So now I have an oxygen to actually form that phosphoester bond. We know that then at really high pH, we lose all of those hydrogens, which then it bonds too much. So we do want to keep it at physiological pH. So we said the second piece to our nucleotide is our monosaccharide. And all nucleotides are going to be constructed from one of these 2 monosaccharides. Our beta ribofuranose, so remember beta meaning the OH is up top here, and a beta 2-deoxyribofuranose. So deoxy, remember, meaning that I've lost an oxygen on the second carbon in our chain. So one of these 2 sugars is going to show up in every single nucleotide that we look at. Lastly, let's take a look at our organic bases. So here's kind of a zoomed in view of each of these bases. So our thymine, guanine, cytosine, and endiosine. Notice that 2 of them look very similar and the other 2 look very similar to each other. I.e., there's 1 ring here, 1 ring here, 2 rings here, 2 rings here. So let's look at that a little bit further. So the one with 2 rings, we're going to call purines. So these are all types of our amines because we've got all of our nitrogens. And when you have 2 rings put together, the 6-membered and 5-membered rings, they're called purines. Then the ones with 1 ring, we're going to call our pyrimidines. So these are our cytosines and our thymine, our C and T in DNA. We'll see that we also have this U, this uracil for our RNA. So these are kind of the 5 organic bases that we're going to be looking at for this chapter. Now, knowing these structures as well is going to help us see why in DNA we know A goes with T and C goes with G. It's because of this structure here that we'll see that they'll be able to form certain hydrogen bonds and not others. So that's kind of where we're going to go with this. So now let's start putting these pieces back together. So when I have a ribose or deoxyribose, and I combine it with one of these organic bases, the purine or pyrimidine base, I'm going to form a nucleoside. So this is only the sugar and the organic base as a nucleoside. So nucleosides containing a ribose are going to be called ribonucleosides. So notice it's only the sugar and a organic base. And here's another one. And another one, and another one. So these are just different organic bases we're putting on each of these ribose sugars. And here's what they all look like with the deoxyribose sugars. So once again, notice I've got the 4 different organic bases, but this time it's a deoxy, since at that second carbon there's 2 hydrogens instead of an oxygen and a hydrogen. Also kind of notice where these organic bases are being connected. I'm being connected on the 5-membered ring, the bottom nitrogen, and on the 6-membered ring for the single cyclic compound, also on the bottom nitrogen in the ring. So notice it's not the nitrogen in between the NH2 and the C double bond O, or these 2 C double bond O's. This one's kind of well hidden in there. It would be very difficult to interact with this one since there's so much stuff around it. So it interacts with the one with less stuff around it, or this nitrogen here. And then just to compare the nucleosides side by side, we know the oxy has the oxygen once again, and the deoxy does not. So just to kind of see it side by side, here's where that oxygen is going away. Now the one that we are more familiar with now are our nucleotides. So the only difference between a nucleoside and a nucleotide is that the nucleotide also has the phosphate group on it. And we said it forms a phosphate monoester, a phosphoester bond. So now I've got this phosphoester bond on one portion of our sugar ring. So the nucleotide has the sugar and the phosphate residues connected by this phosphoester bond. And in the nucleotide, it has the sugar and the organic base, and we're going to say this is connected by an N-glycosidic bond. Remember, a glycosidic bond for 2 sugar residues was kind of the ester residue, or the ether residue. So it had COC for our glycosidic bond between 2 sugar residues. However, now it is a CNC bond. So it's connected at the N, so the nitrogen. Remember how we named our amines, or our amides, we had something connected to a nitrogen, we put an N out front. So an N-glycosidic bond, so a sugar connected at the N of an organic base. Now we can actually attach the phosphate group on different parts of this ring. The sugar residue, since it's a glycosidic bond, or the organic base residue, since it's a glycosidic bond, will always be at the anomeric carbon. So it'll always be at that first carbon after the oxygen in our ring. However, this phosphate bond can be at the 3rd part of the ring, so a 3'. So if it's at the 3' carbon, we're going to call it a 3' nucleoside. But more common in nature, the phosphate is found on the 5' carbon. So if we count 1, 2, 3, 4, 5, and then I put that phosphate bond on there, we're going to call this a 5' nucleotide. And just like before, we can have a ribose, so I still have that oxygen on the sugar ring, or our deoxyribose, so I can take the sugar off. So there's a whole bunch of different ways that we could piece these together and kind of different pieces that we can use for each one. So the phosphate can go on pretty much the 5 or the 3 prime of the carbon. The base, we can add those 5 different bases, so including the uracil. And we can have then 2 different sugars, our ribose and our deoxyribose. So let's try piecing one of these together. So I want you to draw a nucleotide, and I want it to be an andosine 3' monophosphate, and label the phosphoester bond and the N-glycosidic bond. So I want you to attach the phosphate and the organic base onto the appropriate carbon or the appropriate bond in this sugar residue. All right, so let's take a look at what this would look like. So we are going to attach our organic base. We said at that bottom nitrogen in the 5-membered ring, and that forms then our glycosidic bond. Notice that it is going in the up position. So because it was a beta sugar residue, we're going to attach it at the top. So it's basically taking the place of the oxygen. And then we want it at 3', so we add, and it's 1 phosphate, monophosphate. So we're adding 1 phosphate group on the 3' carbon. And here's our phosphoester bond. Since we know an ester is a C double bond O, O, C. So it's a phosphoester because it's a P double bond O, O, C. So I'm replacing that carbon with a phosphorus in this phosphoester. So up to this point, we've looked at the monophosphates, so only having 1 phosphate here, but that's not even the only permutation of this phosphate that we can have. So let's take a look at our di- and triphosphates. So a nucleoside di- and triphosphates are energy rich. They store a lot of energy. And they release a lot of energy when they're hydrolyzed or when those phosphates are removed. So if this is the andosine 5' monophosphate, an AMP, if I add a phosphate to the nucleoside of the monophosphate, it's going to generate a diphosphate. So the way we name this then, it's an andosine 5' diphosphate, aka an ADP, so also known as an ADP molecule. Maybe that's starting to sound more familiar. But I can even add a third one. So adding a third one to the diphosphate, and check out what we have. An andosine 5' triphosphate, also known as ATP. So this is our energy molecule. This is how we get energy, and we transport energy through our body is through this ATP molecule. And we use the energy from the bonds being broken of these phosphoester bonds. So these phosphoester bonds are extremely important in our body because this is when they break and get released, that energy is what we use to survive. That is the energy we use in our body. So they are the immediate source of energy for most of the energy requiring processes that take place in living things, not just in our body, but in living things in general. So we take this ADP, we break it, we release that energy, we get the ATP, a phosphate comes off, I get energy somehow, either by eating or if I'm a plant, by sunlight. And when I get more energy, I ingest more energy, that phosphate goes back onto our ADP and goes back to ATP. So it's kind of a cyclic cycle. I use that triphosphate, it breaks off, I ingest more energy somehow, and then it reforms onto the molecule. So in a more chemical cycle, so we can actually see the phosphates on there, I break that bond, I get the ADP plus that inorganic phosphate. I said I ingested energy if I'm a plant from sunlight or from a animal or us from food, and it gets back on, and I've got a chemical energy again. So it's this chemical energy here that our body uses, and we use food energy or some kind of outside energy to put that phosphate back onto our ATP. Now, another interesting reaction with these phosphates is that we said that it typically can go onto the third and fifth carbons. But what if it does both? So notice that the phosphate can actually connect to both the third and that fifth carbon. And it creates the cyclic nucleotide. So when two phosphoester bonds connected the same nucleoside monosaccharide. So I form a secondary ring with this phosphate group. Now this is an extremely important molecule, and let's look at why. So looking at this process, have you ever wondered how our body actually uses adrenaline? Like we know that we can produce this molecule adrenaline, but what does it do? How does it work? Well, when we produce adrenaline, it connects to this receptor in our cell membrane. The catch is that adrenaline itself cannot pass through the cell membrane. So it somehow needs to send its signal through the cell membrane and into the cell to create that fight or flight mode. So it's what actually ends up happening is what's called the G protein signaling cascade. So the adrenaline binds to this receptor. This G protein here gets the signal from the receptor and it passes it to this adenyl cyclase protein. Now, when it combines to this protein here, this part combines here, this part breaks off. So notice it's orange here and green over here. So it breaks it and it binds to this protein here. Once it binds, it actually ends up creating or telling the protein to create this cyclic AMPs. These cyclic AMPs then, our messenger molecules, they go and tell everything in the cell, hey, we've got an issue. But the way it does that, the way it actually signals, is that they're effectors for allosteric enzymes to regulate these biological processes. So they enable these biological processes to start occurring, such as the breaking of glycogen, so that we create a whole bunch of energy because we're starting to pull off all those sugar residues from the glycogen. So we think of having an adrenaline rush. Not only does your heart start pounding, but you get all of this energy all of a sudden. Well, it's because that adrenaline molecule bound to that receptor created this cascade of reactions, and it sent messengers to start binding to the glycogen molecule or the enzymes that kick off all of the glycogen molecules to create this energy. But that's also after the adrenaline rush, you crash because you've used up so much energy from that cascade that it does take you a while to recover. So you first get this rush of energy because it's all of a sudden breaking all of these energy bonds, and then we crash and get tired. All right, so let's start. These are just single nucleotides that can do all of this. Now let's start looking at what polynucleotides can start working on. So first off, what is a polynucleotide? So nucleotides can be connected to one another from an oligonucleotides, so 2 to 10 nucleotide residues, just like our oligosaccharides 2 to 10 sugar residues, and our polynucleotides, which then is more than 10. So DNA is made from polymers of these deoxyribose nucleotides, so more than 10. And our RNA then is made from polymers of the ribonucleotides, or more than 10 nucleotides put together. So just kind of an overview of DNA, and it's these polynucleotides. They are going to be connected to one another by a 3', 5' phosphodiester bond. So it's diester because each nucleotide has 2 ester bonds. One of them is going to come off of the 3' and the other one is coming off to the 5'. So one nucleotide, it's attached to the 3' and the other nucleotide, it's attached at the 5'. So that's kind of how we create this kind of ladder structure almost. So this is DNA, so it's the deoxy. So notice that each of these sugars do not have that oxygen on the second carbon. Also, we are going to label these as a 5 terminus and a 3 terminus. So it kind of is going to help us figure out how DNA is going to be made because we have a direction for this DNA. So the 3 terminus comes first, and then we go to the 5. So it always comes 3 to 5, 3 to 5. So the 5 is going to be on top, the 3 is going to be on the bottom. So a little bit more about this structure. I also have a sugar phosphate backbone. So we talk about the sugar backbone of DNA. So it's a sugar phosphate, or sugars held together through our phosphate bonds. Now RNA has pretty much the same structure as DNA with a couple of catches. First one, it's R, so it does have that extra oxygen there. So it's not a deoxy anymore, it's just the ribose. So it does have the oxygen. The second difference is that in RNA, the uracil replaces the thymine. So we have a slightly different organic base here. All right, so that is all we're going to look at for this part 1 of our DNA chapter. The next part we're going to look at, delve more into the structure of DNA and how come DNA we know has the double helix and goes up, and how do they know, and all of that cool stuff. So that will be in part 2.